Let's switch gears here and start talking about gene expression. To begin, we need to review the three statements of the central dogma, which reminds us how information flows from the DNA molecule to a functional protein in a living cell. The central dogma tells us that DNA is used to make RNA through the process of transcription. RNA is then used to make protein through the process of translation, and it's proteins that give cells their traits. So when we use the term gene expression, we're talking about this flow of information through the cell, from the DNA molecule to the functional protein. It's important to remember that the first statement of the dogma, that DNA is used to make RNA through the process of transcription, tells us that all RNA is made from DNA. That includes mRNA, rRNA, tRNA, the three primary types of RNA, as well as other types of RNA that are less well known. Things like small interfering rRNAs and other RNAs that we may not have even discovered yet. There's a reason, therefore, that the first statement of the dogma says that DNA is used to make RNA, not just mRNA. Of course, it's only messenger RNA that contains the code that will help build the protein. But we have to remember that both rRNA, ribosomal, and tRNA, transfer, are also required to build the proteins that give cells their traits. The other thing we have to remember, just as we saw with DNA replication, is that the specifics of transcription in prokaryotes versus eukaryotes are different. It's a simpler process in a prokaryotic cell. Transcription is only going to occur in a cell when that cell needs a specific protein in that moment. That means that fundamentally, gene expression is different from DNA replication. We said that DNA needs to be replicated when a cell is preparing to divide. We said that the entire DNA molecule must be copied so that each daughter cell gets her own copy of the DNA molecule. In transcription, the cell has a particular need in that moment for a protein. The DNA is going to be transcribed only in the area of the gene that contains the code for that protein. And that means that the double helix of DNA only needs to be unwound in the area of that gene. Another big difference between replication and transcription is that only one strand of the DNA is going to be transcribed. It's what we refer to as the template strand. Once the DNA double helix has been unwound in the area of the gene and the template strand is exposed, complementary rRNA can be built upon that template DNA. Just as we saw in our DNA replication, RNA is going to be built by combining subunits with covalent bonds, but this time to create a single-stranded molecule. Those covalent bonds between the subunits of our RNA are referred to as phosphodiester bonds, just as they are in DNA. The enzyme that catalyzes this important work is called RNA polymerase. And just as we saw for the enzyme DNA polymerase, RNA polymerase requires an activated form of the nucleotide subunit in order to add it on to a growing strand of RNA. The nucleotide that is going to be added on has to be in that triple phosphate form, just as we saw for DNA. This time, though, instead of being a dNTP, it's just an NTP, a nucleoside triphosphate. So DNA polymerase and RNA polymerase are similar enzymes. They both catalyze the formation of phosphodiester bonds in strands of nucleic acid. Both of them require a nucleotide that has been activated with the addition of 2 phosphate groups. Another key thing to remember is that both of these enzymes can only work in one direction. All nucleic acid is synthesized in the 5' to 3' direction. Let me say that again because that's such an important thing to know. All nucleic acid, whether it's DNA or RNA, is synthesized in one direction, from the 5' to the 3'. And what we mean by that is new nucleotides can only be added to the 3' end of a strand of nucleic acid. Of course, one of the biggest differences between the process of DNA replication and the process of transcription is again that fact that only one strand of DNA is going to be used during transcription, that template strand, the strand that contains the gene. We refer to the other strand of DNA as the non-template strand, and also as the coding strand. The code to build the protein is found in the template strand. The reason we call the other strand the coding strand is because it's going to match the bases in the mRNA, except for the fact that there is U or uracil in the mRNA and T or thymine in the coding strand. So it can be confusing for students to understand that it's the template strand that is going to be transcribed to make messenger RNA. The coding strand is there simply to bind with and stabilize the template strand. The messenger RNA, when it is transcribed, is going to have a sequence of bases that is complementary to the template strand. But when compared to the coding strand, the sequence of bases will be identical, except, of course, for the substitution of uracil in the mRNA, where there's thymine in the DNA. So let's take a look at this. This template strand has the following sequence in a gene. Now, this would be a very, very short gene, and it's not an actual gene by any means. But we can determine what the sequence of bases in the messenger RNA would be simply by performing a complementary match. Remember, there is no thymine in messenger RNA. It's replaced with uracil. However, there is adenine in messenger RNA. So you have to be careful to think as you determine the complementary base placement. Where there's an A in the template strand, there will be a U in the messenger RNA. Where there's a T in the template strand, there's going to be an A in the messenger RNA. Sometimes those bases get confusing. C binds with G and G binds with C, just as we see in DNA. The process of transcription has 3 distinct steps. We refer to those as initiation, elongation, and termination. We're not going to review all of the details of each of these processes in this course, but you should recall the general principles. On this slide, you're looking at a diagram of the beginning of initiation on the left, and then the time just before the end of initiation and the beginning of elongation on the right. The green that you're looking at is a portion of the RNA polymerase enzyme that we refer to as the core. The blue that you're looking at represents the other portion of the RNA polymerase enzyme called the sigma. Now, we use this terminology only for prokaryotic cells, and we use prokaryotic cells as our model when we're learning about transcription, because the process is just simpler in those cells. The DNA molecule is represented in red, and you'll notice that a portion of it is colored blue. The blue represents a portion of the gene in the regulatory region called the promoter. Sigma recognizes and binds to the promoter as the first step of initiation. This binding then helps guide the core of the enzyme into place. The 2 strands of DNA will be separated from each other, and the DNA will start to be fed into the hollow channels of the core. There are 3 distinct channels in the core of the enzyme. One that is going to hold the downstream DNA, one that is going to hold incoming NTPs, and one that is going to hold the growing RNA strand, the new RNA. Once the core is in place, once the channels are located where they need to be, with the correct materials inside of them, sigma is going to release from the promoter and elongation will commence. Remember, there are these structures jutting out into these channels that we refer to as the zipper and the rudder. These are just physical projections of the core that are literally causing the DNA strand to unwind as it passes across them, and guiding the DNA in one direction and the RNA in another. During the elongation phase, the core part of the enzyme is going to be adding the incoming NTPs onto the growing strand of RNA by building phosphodiester bonds. At the end of the elongation phase comes the 3rd phase, which is referred to as termination. Eventually, the RNA polymerase enzyme is going to come to the end of the gene. In other words, it's going to come to the place in the DNA where the code for building protein ends. There's no way for the enzyme to know that it's at the end of the gene. So what happens during termination is the enzyme will continue transcribing the DNA past the end of the gene into a sequence of bases in the DNA that's called the termination signal. All the termination signal is a series of bases in the DNA that contains code that will produce an mRNA that will fold back upon itself, that will complementary bind to itself, forming a hairpin loop. It's this binding to itself that will cause the mRNA to no longer fit neatly into the channel of the core. This process is going to disrupt the core and, in fact, disrupt the entire process of transcription, causing it to end. The process of transcription in eukaryotic cells is more complex than it is in prokaryotic cells. One of the key differences to remember is that there is no sigma in a eukaryotic cell. Instead, there are what we call basal transcription factors, or BTFs. In eukaryotes, there are many BTFs, and the BTFs are what will recognize different promoters and bind to them, allowing for the RNA polymerase enzyme to also bind and transcription to be initiated. In bacteria, there is a single sigma, and it's going to bind to all of the different promoters and initiate transcription. In eukaryotes, there are many BTFs. It's just a much more complex process. You should also recall that promoter regions in eukaryotes are simply more diverse, simply more numerous than what we see in prokaryotic cells. Something else to remember about transcription in eukaryotic cells is that there is not one RNA polymerase enzyme, but three. There are what we refer to as RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. These three polymerase enzymes are simply more specialized than the single RNA polymerase found in prokaryotic cells. Another thing to remember about transcription in eukaryotes is that in a eukaryotic cell, transcription is going to be followed by a rather extensive processing procedure for the RNA. This processing is going to create a mature messenger RNA that is ready for translation. Remember, in a prokaryotic cell, transcription and translation are occurring in the cytoplasm, since there is no nucleus. Transcription and translation occur simultaneously in the cytoplasm of a prokaryotic cell. Eukaryotic cells are different. In eukaryotic cells, transcription is going to occur in the nucleus. Translation is going to occur in the cytoplasm. So the processes of transcription and translation are consecutive in eukaryotic cells. Now let's take a minute and think about this RNA processing that occurs in eukaryotic cells. In the diagram at the bottom of the slide, you can see a drawing of a piece of DNA. This DNA represents a gene. Remember that a gene contains two distinct types of code in it. It contains the code to build a protein, and it contains regulatory code. The regulatory code that is in a gene is going to determine when a protein is made and how much of a protein is made. When transcription occurs and the RNA polymerase enzyme transcribes the protein coding region of the gene, in a eukaryotic cell, a transcript is created that's referred to as a primary transcript or an immature transcript. In this drawing, you can see both dark yellow and light yellow regions in this primary transcript. These represent both exons in the dark color and introns in the light color. Remember, the primary transcript contains some code that is not going to be used when the protein is produced. That is the intron code. Those introns must be spliced, they must be removed, and the exons joined together. Once that occurs, what's left is a spliced transcript. The last part of RNA processing includes the addition of a cap and a tail. The cap consists of methyl 7-guanylate triphosphate, and the tail consists of a long strand of adenines. The cap and the tail are going to do two things. They're going to help during the translation process, and they're going to help protect this mature mRNA transcript from being degraded too quickly when it goes into the cytoplasm.